The first question was how muscle contraction would differ if a skeletal muscle had expressed more calcium pumps than normal. With more calcium pumps than normal, we're going to have a cell that is pumping calcium out of the cytoplasm faster, and so that means that the contraction will end sooner. Calcium is pumped out of the cytoplasm, either out of the cell or into the sarcoplasmic reticulum by pumps. And so that'll remove the calcium earlier. So be pumped back into sarcoplasmic reticulum or outside the cell. Contraction would last for a shorter amount of time because of the reuptake of calcium into the sarcoplasmic reticulum. And the same for this answer. The second question asked how muscle contraction would differ if there were no T tubules. So if here's a cell, a muscle cell, and here are the T tubules. Remember, they're just little indentations. So these are holes down into the cell, like little pits or wells, and they allow the action potential to both go around and further, but they also go down into the cell. That means that sarcomeres that are near the surface would get calcium from sarcoplasmic reticulum, opening voltage-gated calcium channels and allowing the calcium out and into the sarcomere, but also the ones deep in will have voltage-gated calcium channels from sarcoplasmic reticulum that when the AP reaches them, that will open these voltage-gated calcium channels and we'll get calcium out. So if we get rid of these T tubules, then we still have a chance of getting calcium in from the surface. If there are voltage-gated calcium channels here at the surface, we can get calcium into these sarcomeres and get contraction, but these deep ones won't get any. And so we'll get a weaker contraction, either without it or a weaker contraction. So calcium won't be released deep in the cell to cause the muscle contraction. I want to correct a potential misconception. I've had quite a few people say unless the action potential is really, really strong, but that wouldn't be the case. All APs on a specific muscle cell or neuron are essentially identical. That's true until the cell starts to get weak, and if it gets that's true unless the cell starts to lose its sodium and potassium concentration gradients. So if it can't make more ATP, the sodium-potassium pump will stop, and you could get slightly smaller APs as the concentrations decline. But under any normal circumstances, they are all identical. Third was a sketch showing an extensor and flexor for the arm. So here's the humerus, your upper arm bone, and radius and ulna. They work together, but they're both long bones. And here is the joint at the elbow. This is I think it's the olecranon process. It sticks out at the back of your elbow. And you have a muscle on the back of your arm, the triceps, that will pull up. Remember, muscles always shorten, and so that's what happens to your arm. And the biceps, which pulls up here, and if the biceps shortens, the arm flexes. Here's how your knee works. You actually have both the extensor and the flexor are on this part of the thigh. The flexor is attached here and here. So like this blue, although it doesn't actually go quite that far down, but if we contract this, your knee will bend. the extensor is actually contracted to your kneecap, a small bone, which then has a tendon down to your shin bone. And so when you contract the extensor, that pulls up on your patella, that pulls up on your leg, and as your patella shifts position, you pull the leg up. And here's one showing essentially the same thing, the biceps doing the flexing, and the triceps, when it contracts, doing the opposite. And at the knee, same thing. Again, it would be attached to your kneecap, right, not actually directly to your shin bone. We'll go through in class a way to get neural circuits showing different sensory neurons in the muscles or tendons to detect, one, a sensory neuron detecting that a muscle is shortening, and two, that it's lifting a very heavy weight. So there were some good answers here. In class I'll talk about the actual ones, but this is in fact what happens for A when the muscle shortens. There are stretch channels in the dendrites of a sensory neuron here, and those stretch channels allow sodium in. You get action potentials when the muscle goes from down here to up here. This one was a clever idea that maybe when you have lots of, when you're trying to lift a heavy weight, you have lots of neurotransmitter release from the motor neuron. And so here's the motor neuron releasing acetylcholine to the muscle, and that's causing muscle. Maybe there are some acetylcholine receptors on a neuron nearby, and they're detecting the really high amounts of acetylcholine. So the amount of acetylcholine getting here would affect the number of action potentials. It's not actually the way it works. The way it works is actually a little simpler, that in this tendon, so here's a tendon here, and there'd be a tendon up here, tendon down here. In this tendon, there would be a sensory neuron going back up to the spinal cord. And the sensory neuron in the tendon has these dendrites that are wrapped So the dendrites would be wrapped in here through the tendon, and when there's a high strain, that puts pressure on these tendons, and it opens stretch channels. So to detect a heavy weight, you have stretch channels in your tendons, and those are when the muscle is working really hard. It's pulling, there's a lot of force on the tendon, it's pulling hard on it, there's a weight that's pulling it down, so you get a lot of action potentials per second, and that tells you it's a heavy weight. Very few action potentials per second, and maybe you're trying to lift a pencil. Here's another one. This one did use stretch channels in the muscle to detect when it is shortened, and that is how it works. And then over here, as the muscle works hard, you build up lactic acid, and so maybe some metabolite that's a metabolic byproduct of heavy work, maybe that binds to a receptor, and maybe that activates the neuron to tell you that you're lifting a heavy weight. But in fact, it's just stretch channels in a tendon. For number 5, a neural circuit showing an extensor and a flexor at a joint in an insect or crustacean leg. So that's an animal with an exoskeleton. And so here is the exoskeleton. Okay. So the exoskeleton, and the extensors are this one, because if we pull here, if we shorten A, then this moves, then this moves like so. So this moves this way, this end moves this way, and the leg is going to extend. And so A is an extensor and D is an extensor. Here's the pivot point for A and the pivot point for D. B is a flexor. If we pull on this, it's going to pull this way. So if B shortens, we're going to flex. We're going to bend this limb this way. And if C contracts, we're going to flex. We're going to bend this last part of the leg this way. And here are the neurons that control them. For B, for C, for D, and A, and the motor neurons could go to any one of those. What if there was an injury? Well, if muscle A was injured, so we've lost muscle A, then maybe it couldn't contract anymore, and therefore the organism couldn't change between these two positions. So if A is not working, this whole part can't extend anymore. So here it is bent. Here's where A would be. If A is not working, you can't go from this bent, you can't do that.